Good morning, everybody. Welcome to day 3 of the 2025 ISS Research and Development Conference virtual technical sessions. Really glad to have everybody here today. We have another great set of presentations for you, some really very interesting research, and our presenters are looking forward to your questions. So in case we didn't meet over the past couple of days, I'm Jim Way. I'm Executive Director of the American Astronautical Society. Our organization has been responsible for assembling these technical sessions for the ISSRDC for the past several years. And we've sincerely enjoyed our collaboration with everybody at the ISS National Labs on this, as well as teams from NASA and other collaborating partners. We're looking forward to what comes next with all of this. We know that a lot of the low Earth orbit talk will be a part of AIAA's Ascend, which will take place in May in 2026 here in Washington, D.C. So be sure to keep your eyes open on that one and look out for registration, and we hope to see you there. So let's get right into it. And so without further ado, let me introduce Philip Iris. He's Program Director at the ISS National Labs, and he's our session chair this morning. Philip.
All right. Thank you, Jim. Good morning, everyone. Welcome to our first session of the day. This is going to be focused on biotechnology and medicine. Hopefully you West Coasters are on your first cup of coffee, while us East Coasters have probably already had two. As Jim said, my name is Phil Iris. I'm a Science Program Director with the ISS National Lab, and I'm going to be your session chair. So here's how things are going to go. First, we'll present the 5-minute executive summary video, and then we'll have that author come on screen for a 5-minute live Q&A session. We'll then move on to the next presentation and author, and so on. Webinar attendees will not be able to come on screen, so please use the Zoom Q&A tool to submit any questions you might have. Please don't be shy. We want to have a nice, fruitful discussion with all the authors, and I think that has been the case the past couple of days, so I expect it to continue. If we have time left at the end of the session, we can address any outstanding questions that have come up along the way. Lastly, all the sessions are going to be recorded, and they'll be made available on the website as soon as possible. And if you want to see the full 20-minute presentations, please visit the online program and open up that file. So without further ado, we'll go ahead and get started with our first presentation. We have Fatih Karui, a senior research scientist and in-space commercialization lead for the Blue Mountain Space Institute, and also affiliated with Bioserve Space Technologies. So we'll go ahead and show the video.
Hi, my name is Fatih Karui. I am based at Blue Mountain Space Institute of Science at NASA Ames Research Center. I'm talking to you today about the microbiome and the metabolome of the International Space Station. As you can imagine, it takes a talented team to be able to conduct this type of study, and so this was done under the leadership of Peter Dorenstein, as well as Rob Knight from the University of California, San Diego, and Katsuri Venkateswaran from the Jet Propulsion Laboratory. So 3D stands for 3D microbial mapping, and so the idea here is to map the microbiome as well as the metabolome from the USOS, which stands for United States Orbital Segments. For this, we use double-headed swabs, so we collected over 800 samples from the ISS, and so one head of the swabs was used for nucleic acid extraction, subsequently 16S and metagenomic sequencing, and then we're doing the analysis. In parallel, we look at actually metabolites, and so we did some metabolite extraction using untargeted mass spec to kind of be able to characterize the metabolome. In this graph, kind of highlighting specific features related to how we perform the analysis from a QC perspective, and so the microbiome here and the metabolome. So here is highlighting a specific map of the ISS, and more specifically all the different modules and the numbers of samples that were collected. And so node 3 stands for the module where you have the waste and hydrogen compartment as well as the exercise machine. PMM is a storage room. Node 1, that's where you have a dining table. Laboratory, that's where most of the science is conducted. Connected to node 2, where part of it is science related. The other is kind of crew quarters where the crew sleeps. Node 2 is connected to Columbus module, which is European contribution, and which is also a laboratory. And Node 2 is also connected to the Japanese module. which also has a logistic module where storage is located. So one key features of analysis is to conduct a 3D visualization. And so, for example, here I'm highlighting Micrococcus, which is actually a fecal particulate or fecal matter. And so to do that, we kind of look at that particular species and we kind of compare it to a reference organism. And so as you can see here, there's a specific features looking through at the node 3. And so as you can see, we were able to see some particulates surrounding the western hydrogen compartment and kind of located also next to the western hydrogen compartment. But as you saw, there was also some features outside of that compartment, and so certainly this is of concern. From a metabolite perspective, here is kind of emphasizing the fact that 95% of the feature actually are not found in existing library, which means that only 5% of the feature could be annotated from existing laboratory, and so which is pretty remarkable. Here is kind of highlighting all those particular annotated features based on their location. What was pretty interesting is that node 3 as well as node 1 had particular unique feature as opposed to other areas within the ISS. So here also I'm visualizing specific compound, and in particular here we're looking at hippurate acid, which is a urine toxin. And so kind of originate in node 3, where you have the western hydrogen compartment. But as you can see, similar to fecal matter, you can see some trace of that particular compound outside. But when you do like a molecular network, you can see that its derivative also could be found in other area on the ISS. When we look at, for example, a food-related metabolite, in this case, piperine, and so, and here I'm highlighting node 1, which is a node where you have a dining table, which is highlighting here, and so you can see specific features kind of surrounding the table, but also above the table. And again, what was nice to see is that actually that particular metabolite was only found in node 1. So conclusion from the study, so this was the largest study by 100x compared to what was done in the past. More importantly, also it had a metabolome contribution, which was the first time to be able to do so. And what was also interesting is that each module's function was derived by the type of activity, and so that could be seen from a microbial as well as from a metabolome perspective. Again, we didn't study the Russian segments, but certainly this type of study kind of emphasized specific highlights about maybe how those commercial platforms should be built in the near future. And so certainly this was pretty remarkable to kind of see that there was a significant decrease of microbial diversity and an increase in xenobiotic compounds. Again, this was done with help from Kate Robbins, which was the one which was actually collecting the samples. And then also we had some help from Michael Barrett, which helped us with some aspect from the logistics and from the planning perspective. And so here are also all the individual and supporting funding from the ISS National Lab. Thank you again.
All right, thank you, Fatih, for that great presentation and welcome.
Thank you. Good morning to you.
Yeah, good morning to you too. So we'll go ahead and wait for a couple questions to pop up in the Q&A. Here's the first one. Were you able to distinguish human origin species from other animal origin species?
Yeah, clearly we were able to differentiate all the different community. As you can imagine, the bulk from the environment when we were collecting surface samples, the bulk came from a human, but in particular skins. And so that was more or less the majority of the origin from the source tracking. But yeah, you do see variations, which, again, belong to the environment, human, animals, plants, and etc.
Okay. I also saw you had that only 5% had a library match. Can you expand a little bit on what the other 95% is or might be?
Right. So, again, I think that derived just from the fact that we're still lacking data from a metabolomic perspective. So the bulk of the data from a metabolic perspective is kind of related to human as a host and disease in some sense. Here we're just emphasizing on the environment, and so that's the reason why only 5% of the metabolite that were detected could be found on those specific databases. So hopefully, over time, I mean, that number will increase all the time, because then we will have more information from the built environment in other locations and etc. But certainly, that was striking to see that only a small amount, 5% again, could in some sense be identified. And so,
Okay, thank you. Quite a few questions popping up. Were you able to determine efficacy of cleaning protocols used?
Yeah, I mean, I didn't show, I guess, yeah, during the presentation, the 20 minutes presentation, I show when the crew was using wipes to kind of clean up through the station. So it was pretty clear that the crew is doing a great job. And actually, the station is too clean. From a microbial perspective, as I mentioned at the conclusion, I mean, one of the key takeout from the presentation is that if you compare the built environment on the ISS to other type of closed habitats and outside like housing, hospital, and etc., the ISS is on the far hand from a diversity perspective. And we know very well that there is a strong correlation between human health and being exposed to like a healthy microbiome and a healthy microbiome of the built environment. And so certainly in our situation, we clearly see a lack of diversity on the ISS. And so probably the station is overly clean from a health perspective.
Okay. Is there a possibility of on-orbit sequencing for microbiome analysis?
Say again?
Sorry, is there a possibility of on-orbit sequencing for microbiome analysis?
Yeah, it is already ongoing. I mean, that has mainly been spearheaded by NASA Johnson Space Center, where they're leveraging mini ions as a sequencing platform. And so, as you know, for a very long time, monitoring on board the ISS was mainly culture-based. And so now I guess you can leverage using mini ion to kind of try to identify and characterize the environment. And so, again, it is still complex and complicated to kind of process all those steps. Nevertheless, that was tested and validated not too long ago. And so, yeah, that's certainly something that down the line, I mean, when we look at longer kind of class missions, certainly that would be something nice to have, where you can do some monitoring based on molecular-based analysis. Nevertheless, I mean, if I may add, it's still very important to do culture-based because, again, then you are fully able to characterize those microorganisms from a phenotypic point of view and physiological point of view, which is also key.
Okay. Next question is, what kind of instrument did you use and what challenges did you face during experimentation?
So, I mean, we didn't use instrumentation per se on board. So, as you know, I mean, despite the fact we call the ISS a laboratory, I mean, there's still not much in situ capabilities to do analysis. I think the challenge for us is that this payload was implemented during COVID. And so actually we weren't able to purchase those swabs. And so we had to 3D print like 1,500 of those double-headed swabs, and then having the students kind of build them hand by hand. And so that took a lot of work for sure, but certainly, yeah, no particular analysis was done in space. Everything was brought back to Earth, and then we conducted the analysis on Earth at the University of California, San Diego.
Okay, we'll do one more question here. Are there plans to conduct periodic testing to see how the microbiome distribution changes in the ISS over time, such as during crew exchanges or new supply arrivals? And I imagine it'd be interesting to study on the CLDs, right, from the start of a space station as it evolves into a fully operational system.
Yeah, definitely. I mean, that has been something that NASA Space Biology program has spearheaded over the past few years. And so it's part of the microbial tracking series. And so actually I was lucky enough to manage the first two missions, which was microbial tracking 1, which was only environmental. Microbial tracking 2 was looking also at the environment and then the astronauts. And for those two studies, actually, we did multiple sampling over time. And so you were able to actually see how the microbiome of the built environment was changing over time. What was also interesting when you were doing those type of having like time points, actually you could see and pretty detect that you could identify an organism maybe like only in one location, When you came back a few months later, then you realize that that organism was now in all locations on the ISS. And so certainly there's added value to be able to monitor the station on a regular basis. And certainly from a commercial perspective, I mean, that would be good to do, but more importantly, I think one of the probably outcome from the conclusion from a microbial perspective is that probably you will want from those commercial platforms having the waste and hydrogen compartment on one hand of the station, as opposed to be next to where you exercise, next to where you get access to the cupola, and etc. Because you clearly saw that actually fecal matter was also outside the waste and hydrogen compartment. And so probably one of the recommendations would be to have like a zooming in some sense from those commercial platforms, but definitely, sampling on a regular basis would be very, very helpful.
All right, well, thank you very much for your presentation and your time. And if you'd like to continue answering these questions that are popping up in the Q&A, feel free to do so through the chat.
Thank you.
Anything else on that front, Jim? No? All right.
No, all good. Yep.
Okay, so with that, we'll thank you again, and we'll move on to our next presentation. Next, we will have Catherine Martin, Digital Initiatives Lead for Mini PCR Bio, and her talk is titled A Decade of Discovery, Celebrating 10 Years of Genes in Space and Student-Led Achievements in Space Biology. Here's the presentation.
Hello, and thank you for joining me. My name is Dr. Katie Martin, and today I'll be speaking with you about the Genes in Space Student Research Program. Genes in Space celebrates its 10th birthday this year, and we'd like to use this opportunity to look back at some of the moments of achievement that have happened over the last 10 years and look forward to what we hope we can accomplish in the next decade. At its core, Genes in Space is an ideas contest. Each year, we challenge hundreds of U.S. students in grades 7 through 12 to think creatively about the challenges of space exploration. We invite them to identify an open research question, propose a hypothesis, develop an experimental plan, and explain how their results will impact the future of space flight. The winning team gets the opportunity to send their experiment to space to be conducted by astronauts. In this photo, we see 2022 competition winner Pristine Onuoha watching her project grow into something so much larger than she could have dreamed. This is what inspires students to apply to Genes in Space. There's a myth that space research is accessible only for NASA engineers and elite scientists. Photos like this one challenge that myth of inaccessibility. The Genes in Space program was designed specifically to give young scientists the agency to tackle the most pressing spaceflight challenges of our time. It started in 2015. The Boeing Company, in partnership with Mini PCR Bio, decided to address the technology barriers to molecular biology research in space by sending this small, energy-efficient, and easy-to-operate polymerase chain reaction, or PCR, thermal cycler to the ISS. PCR was used widely in Earth-based laboratories every day, but at the time it was untested under the unique conditions of space. But what experiment would be the right one to validate PCR in microgravity? Together, Boeing and Mini PCR made the bold decision to hand the reins of that scientific investigation to students. They put forth a challenge to students: design a PCR experiment that would validate this new tool and enable a new era of DNA research in space. This was the inception of the Genes in Space contest. Anna Sofia Bogorayev was the winner of that first contest. As a junior at Fox Lane High School in New York State, she was interested in studying the immune deficiencies that astronauts experience in space. She learned that when astronauts go to space, their immune systems don't work as well as they do on Earth, and some of those deficiencies persist even after they return. She was wondering if there was an epigenetic reason behind that deficiency. Epigenetics refers to the way genes can be turned on or off by molecules that bind to the backbone of the DNA molecule. Anna Sofia devised a PCR-based experiment to explore whether some immune genes were turned off when they should be on, or vice versa. This is a great research question, but to answer it at that time, the state of space biology research was such that Anna Sofia would need to first validate the tools before she could address her hypothesis. She would need to show that PCR works in space before she could use the technique to tell us something about how space impacts our DNA. We launched Anna Sofia's experiment in 2016, and it became the first PCR experiment ever to be done in space. Not student experiment, but any experiment. Now, 10 years later, Anna Sophia is training to become a physician scientist and earning her MD PhD through a joint program through Harvard and MIT. She's still fascinated by space medicine and regularly involved with the Genes in Space program. And our contest has grown as well. Here are our last 10 years by the numbers. Since 2015, we've reached almost 15,000 students through our Lab in a Box classroom biotechnology loan program. These students have learned how to use biotech hands-on. More than 10,000 students have submitted proposals to the Genes in Space contest. And we've received proposals from students from 48 states and territories, including Alaska, Hawaii, Puerto Rico, and the Northern Mariana Islands. We've launched 11 student-designed experiments to space, and we've published 9 peer-reviewed articles in scientific journals, all of which were first authored by high school students. 10 years ago, it was unclear how much Genes in Space would grow and impact space biology research on the ISS. We accomplished this success by standing apart from other STEM programs and thinking big about the impact we could have, not just on the space industry, but on the STEM workforce writ large. In closing, we'd like to extend a heartfelt thank you to our founding partners at Boeing and Mini-PCR Bio, as well as our sponsors at the ISS National Lab and New England Biolabs. Thanks for your attention.
Hi, Katie. Thank you. Welcome, and appreciate the great talk.
Hey, thanks for having me. It's great to be with you guys.
Yeah, absolutely. We already have questions popping up. So kick it off with amazing program. What type of genes are used in the experiments?
Variable. It really depends on the student's investigation. And over the years, we've even widened the net to allow for like other types. Students can propose any biology investigation. Usually they're talking about DNA, but they can sometimes talk about other molecules entirely, like RNA. So it really depends. I think the experiment we launched most recently that I didn't speak to in the presentation was an investigation on something called phage therapy, which is a really cool idea. It's based on the idea that you could use viruses as an antibiotic. So in nature, viruses are kind of predators for bacteria. They kill bacteria. So the idea is maybe humans, when we're trying to treat bacterial infections, can we weaponize viruses against the bacteria that are infecting us? And this is kind of an important area to explore, especially as antibiotic resistance becomes a bigger and bigger problem for public health. And this would be really applicable in the space setting because, of course, we need to have antibiotics at the ready, and we need them to be kind of efficient and space friendly. And so the most recent investigation we launched is just testing this idea of, can we manufacture these bacteriophages in space? And so that's one example of the genes that we target, but it's like, that's just one investigation. And then the students the previous year looked at something completely different.
Okay. Well, I mean, given the success of your program, right, which is very impressive in the slide you showed, what's your vision for the future of genes in space? Expanding, I see a couple things in the chat about, would you also support international students? Isn't it only targeted to high school students now, right? And I think those are opportunities to expand. So just interested in how you see things growing.
Yeah, I can talk about a couple directions we've thought about. So in terms of international reach, that is something we talk about every year. In the past, we have offered international competitions. We did one in the UAE. We had a competition just for UAE students. And we launched that experiment, I believe, in 2016, that winning experiment. Since then, we have not put on another international contest, but sometimes opportunities pop up and we discuss them. We haven't done it yet, but it's something that we would love to do. And our partners at Boeing, I know, have thought about that extensively. So that may be for, in something we'll explore further in the future. Another thing we talk about a whole lot is, how can we cast the net wider? Like we can launch only one experiment to space each year, but we always are thinking about, like, well, could we name 5 finalists in our contest. We picked the winner from the 5 finalists, but we've thought, like, maybe we could launch all the finalist experiments, or how could we maybe expand our reach in terms of the investigations we're doing? No active plans to do that, but that's something we've thought about. And then in the same vein, I mean, we receive hundreds of submissions each year. And so we've thought about how can we make sure those students are having the best experience possible. They have these great ideas and we would launch all of them if we could, but it's just not possible. But that's what gave rise to our biotechnology loan program, Lab in a Box. So this is a way that students can take their idea from Genes in Space and start to learn about the hands-on technology that we would use on the ISS to implement it if we were to launch their experiment. So that program, we send biotech equipment into classrooms and students can actually use it to do a lab and get that hands-on training. And in recent years, we've expanded that program. So now we're training more students each year through Lab in a Box than we were before. And that gets us super excited. I'm on the mini PCR bio side of Genes in Space. We really care about making science accessible to students. And so that's something that we're super proud of as well.
Okay. Now I imagine the opportunity to launch more and expand gets greater as more platforms go up in low Earth orbit. So
yeah.
Several more questions in the chat. Let's see. Thank you for your presentation. Do you use only PCR in your experiments or anything else?
No, that's a great question. We started as just a PCR contest. So the first Genes in Space contest, like what should we do with PCR in space? Since then, we've launched, as you just mentioned, Philip, other platforms to space. So we have something we call the Genes in Space Fluorescence Viewer. This is a way you can visualize fluorescent biomolecules. So you can label DNA or proteins with fluorescent molecules, and then you can visualize them in this viewer. The past several experiments we've launched have incorporated the fluorescence viewer. And we also have added to our kind of toolkit something called BioBits. This is cell-free technology. So basically, in order to make proteins, which insulin is an example of a very useful protein. So it's a class of biomolecules that's very valuable to have on hand. But traditionally, manufacturing those proteins has required that you grow them in cells because living cells are what make proteins. It's very onerous to keep cells alive. And so BioBits is a way of kind of shortcutting that requirement, making proteins without living cells. So we've launched that as well. And I think our first use of that was our 2021 winning experiment was the first use of BioBits technology in space. And then we've had a couple others that we've launched since that also use that platform.
Okay. I think that's all the time we're going to have for questions. I guess I'll ask you one more. It's probably the most important. And I see a lot of compliments in here about how great your talk was and inspiring.
Oh, thanks.
How can they reach you?
Oh, you can reach, so if you email genesinspace@minipcr.com, that's where you can reach our the whole staff of our Genes in Space program. And my name's Katie. I'm Katie@minipcr.com, Katie with a Y, if you'd like to reach out to me personally.
Okay. And I imagine a lot of those details are on the longer presentation video online. So check those out. And in the meantime, Katie can answer your questions through the chat as we move on. So thank you, Katie. Good to see you.
Sounds good. Thank you guys so much for having me. Great to see you.
All right. Our next talk will be Scott Nagel, a scientific advisor for the National Stem Cell Foundation. So go ahead and play the video and then have a Q&A with Scott.
Greetings, I'm Scott Nagel, Scientific Advisor for the National Stem Cell Foundation, led by Dr. Paula Grisanti. I previously led research at the New York Stem Cell Foundation, where I helped develop programs to study neurodegeneration. Today, I'm presenting on behalf of our broader team, which is building a platform that uses microgravity to better understand disease mechanisms. I'd like to thank the ISS National Labs and the AAS for the opportunity to share our work today. In addition to physiological changes like bone loss and muscle atrophy, microgravity also impacts the brain, including changes in protein aggregation, viral reactivation, and elevated inflammatory signals, which may be relevant to the mechanisms of neurodegeneration. However, brain tissue and cells from living individuals are not directly accessible. Instead, to better understand the complex interplay among brain cells and disease-associated molecular changes, our team developed brain organoids assembled from human-induced pluripotent stem cells to rebuild neural models of disease. From the iPSCs, we can create cortical organoids containing the neurons and associated cell types to model diseases like MS or AD, and dopaminergic organoids containing neural cell types from the midbrain relevant to Parkinson's disease. We assembled our organoids to include microglia, the brain's resident immune cells, which are key to studying neuroinflammation. To improve reproducibility and experimental scale, we also developed a novel cryovial culture approach, housing one organoid per vial, maintained at body temperature and without media or gas exchange. We first used this method on SpaceX 19, where the organoids spent a month in orbit and were successfully returned for post-flight assays. We used bulk RNA-seq to study how microgravity affects cell differentiation. Overall, we found changes in cell proliferation and maturation markers across organoid types, but the effects depended upon their developmental stage. In dopaminergic organoids, early progenitor markers were suppressed, while proneural and mature markers increased. In cortical organoids, cell cycle and early stage markers were reduced, favoring more advanced neuronal and glial cell types. To date, our group has completed 4 additional studies using our cryovial culture strategy, including SpaceX 24, 25, AX3, and SpaceX 30, from which we generated additional high-quality transcriptome datasets. For our SpaceX 30 mission, we collaborated with BioServe Space Technologies to implement a fluidic platform that allowed us to perform media exchange and drug challenge experiments on board the ISS, generating additional transcriptome datasets and imaging data. These platforms and datasets help us learn how to adapt to sources of variation that were outside our direct control. Over the 6 years of developing these models, sequencing methods and vendors changed and launch schedules shifted. The developmental stage of the organoids at launch also varied, but overall, microgravity exposure was remarkably consistent relative to organoid growth within about 3 days, except for AX3, which by design had a shorter stay on station. Meta-analysis across the missions performed by Jim Mossman and our team indicated that despite these variables, common pathways were enriched in microgravity. Interestingly, pathways associated with metabolic alterations and cell proliferation and growth were consistently altered. Around this time, Pierre van der Hagen's group developed a genetic birth dating system to track how human and mouse cortical neurons develop. They focused on mitochondria, looking at their structure, gene expression, and metabolism. What they found was striking. In humans, mitochondrial maturation takes months or years, but in mice it happens within weeks. And by manipulating mitochondrial metabolism of glucose through the TCA cycle and through the electron transport chain during oxidative phosphorylation, with drugs or genetic tools, they could speed up or slow down neuronal development, showing that mitochondria are active drivers of how quickly neurons mature. We thought that insight might be relevant to our work in microgravity, where mitochondrial function appears to play a central role in how cells respond to space-like conditions. During our SpaceX 30 mission, we delivered a similar drug cocktail to similarly manipulate neuronal metabolism. We conducted parallel media exchanges and imaging sessions alongside astronauts Mike Barrett, Laura O'Hara, Tracy Dyson, and Matt Dominique on station. On the right is one of the images we were able to capture during flight of an organoid transduced with a reporter encoding expression of a genetically encoded calcium indicator when a neuronal marker synaptin is expressed. Our drug challenge studies showed that our cortical organoids indeed did respond strongly to LDHA inhibition in microgravity, while dopaminergic organoids showed a weaker microgravity effect upon drug exposure. This coincided with LDHA gene expression, with cortical organoids showing a lower expression that was stimulated in microgravity, while dopaminergic organoids had an already high level but smaller differential expression in LEO conditions. Using gene network co-expression analysis, we have identified subsets of genes whose compound concentration effect was opposite in different microgravity conditions and also consistent across organoid types. This suggests our platform is sensitive to detect drugs that have countermeasure properties to microgravity-induced alterations. And finally, I just want to thank our bicoastal space team and our funders for the support and collaboration in developing this project. We look forward to continuing progress with our NSPA award, and thank you all for your time today.
Thanks, Scott. Welcome, and thank you for your very impressive talk.
Good morning. Good morning. Thank you.
Morning. Well, we already have some questions popping up in the chat, so let's get right to it. What is the effect of fluid flow, or in microgravity, lack thereof, on neural organoid genetic expression?
Yeah, I think we're just starting to beginning to understand, what's going on in these cultures. What we believe we see going on is a difference in the way that the organoids are responding to lack of convection that you have in these cultures on orbit. Possibly, just condensing or concentrating all of the molecules that are being secreted by the cells in the organoid, or building up around those organoids. So that's one of the key things that we're interested in understanding is how those changes might improve our ability to see biological differences in the cell models that we're building.
And next question we have is, great talk, very inspiring. Oh, sorry, that's not your question. Mitochondrial maturation is key to energy production to cells. Have you tried this experiment with any ketones, such as BHB rather than glucose?
No, not yet. We're interested in sort of building on these observations as well. We thought it was interesting that the LDHA inhibitors worked really well, sort of as previously seen in human and mouse cortical neurons, but they didn't have as big an impact on dopaminergic neurons. This is sort of interesting that it could be very cell type specific and brain region specific response to various metabolic components. So we're interested in sort of expanding on that and trying to test other metabolic convergens as well.
Mm-hmm. And we have, how do you ensure that controls on the ground are valid given the environmental differences?
Yeah, this has been a challenge, and one of the challenges that we've tried to address, assembling these experiments over the 6 missions. The cryovial cultures are very simple. They're easy to control. You can set up a single batch of organoids and divide them between the cryovials that are going on ground and in parallel on Earth. So all of these studies have had sort of contemporaneous ground controls in addition to the ones going up on station. For the more challenging manipulations that we did in SpaceX 30 with the chambers from BioServe, we very carefully we had them offset by about 24 hours. And we would watch what the astronauts did on station, watch all of the various procedures that they had lined up to do to change out the media and do the various manipulations on station. And then we would go back to New York, go into the lab and basically replicate the exact same thing on the chambers that we had in the parallel experiments going on ground. So we did as much as we could to make sure that the ground controls were as well matched to the LEO experimental samples as we could possibly get.
Yeah, not always easy though, right?
Yeah, lots of challenges.
All right, let's try one more. Have you considered performing single cell RNA sequence in future experiments, and how?
Yes, yeah. We're really interested to see the bulk RNA-seq data is giving very sort of detailed insights, but it's very hard to distinguish, the activity of individual cells from those bulk data. So we're really interested in applying the various sort of advances in single cell sequencing technology that's come along in the past few years to the organoids that we're going to be setting up in the INSPA program.
All right, great. I think we have time for even one more, and then you have several more in here that you can answer in the chat
Great.
afterwards, and a couple of requests for your contact information. So let's go with the importance of neurovascular coupling. Do you have any plans to incorporate vascular cells into your organoid models?
Yeah, we'd love to do that in the long run. We're sort of starting simple using the organoids with the immune cells that are in there, but over time we'd like to see what the vasculature, the way that has an impact. That we know that there's, particularly in neurodegenerative diseases, there's a lot of interaction among the blood-brain barriers, all the cells that make that up, and the cells that surround them. So we're interested in eventually developing these models to model that as well.
All right, great. Well, thank you very much, Scott. I imagine your contact information is on your full presentation.
I think so. If not, I'll put it into the chat. Yeah.
Okay, I'll put it in the chat. All right, great. And again, feel free to answer those outstanding Q&A questions in the chat. And we appreciate you giving your talk today.
Thank you.
Thanks. All right, our next talk is going to be Maxwell Landolina.
Actually, sorry, Phil, he couldn't make it today, so we're going to skip over his presentation and go right to DeCarlo.
Okay, we'll go to our last talk of the day then, and then we can answer any other outstanding questions at the end. We'll have Roberta DiCarlo, postdoctoral associate with the University of Pittsburgh. The talk is titled "A Novel Mineral Organic Bone Adhesive Promotes Localized Calvarial Bone Regeneration in Microgravity Conditions." Here's the presentation.
Good morning, everyone. My name is Roberta Di Carlo, and I work as a postdoc in the University of Pittsburgh School of Dental Medicine. Today, I'm going to show you my project with the title of "A Novel Mineral Organic Bone Adhesive Promote Calvarial Bone Regeneration in Microgravity Conditions." Everybody knows that exposure to conditions of microgravity or reduced gravity can lead to different complications in normal physiological functions, including bone and muscle atrophy, cardiovascular dysfunction, and reduced immune function. Also, microgravity can alter the musculoskeletal physiology of the astronauts, leading to accelerated bone loss. Latest reports indicate also that such bone loss is not fully recovered even 1 year after return from a long-duration space flight, with poorer recovery associated with longer space missions. Altogether, this evidence indicates that a solution to bone loss and to the inability to regenerate bone must be found before starting long-term space missions. With the aim to find a solution to this problem, and thanks to a Casey-sponsored research program, Rodent Research 25, and the collaboration with RevBio, we completed a series of investigations on board the International Space Station, testing an innovative biomaterial developed by RevBio. RevBio has developed a potential solution for repair of bone fracture and regeneration of bone defect, based on a novel injectable mineral organic bone adhesive called tetronite. Tetronite is a unique combination of multiphasic calcium phosphate and phosphoserine, which offer the opportunity to perform minimally invasive bone regenerative procedure while providing mechanical stability by means of its persistent adhesive bone strength. To demonstrate our hypothesis that tetronite can promote the bone regeneration of a critical size defect by stimulating skeletal stem cells in mice that spend time in space, we introduce a mouse model in which we perform a 2 mm defect, and we compare the ability of tetronite bone adhesive to the one of a well-known osteoinductive biomaterial that is based on the combination of human recombinant BMP2 with a collagen membrane, and these BMP2 serve as a positive control in our experiment. And then we have defects that were left empty without any treatment as a negative control. On the left side of the slide, you can see a timeline where we describe the time where the mice arrival in the animal facility until the day of the launch. Of course, the experiment conducted in space were replicated on Earth to allow for comparison of the efficacy of tetronite and BMP2 to regenerate bone in conditions of both microgravity and normal gravity. After 60 days in space and on Earth, samples were collected, and to evaluate bone regeneration, we performed microcomputed analysis and histological analysis. Regarding the micro-CT, as you can see in the figure on your right, measurements were made in 3 adjacent concentric cylindrical and toroidal zones, mutually exclusive of each other, the defect zone in green, the surrounding zone in yellow, and the peripheral zone in blue. And the defect zone has a diameter of 2 mm, while the surrounding and peripheral has a wall of 1 mm each. A zone equivalent to the totality of the defect zone, surrounding zone, and peripheral zone was mirrored on the sagittal plane and identified at that not surgical left parietal bone as a contralateral side. Okay, so in our study, to assess whether the tetronite bone adhesive is effective in space as on Earth, we conduct both quantitative micro-CT and qualitative histological analysis. Our data show no statistically significant difference in bone regeneration between spaceflight and Earth cohort for both tetronite and BMP2 treatment, meaning that tetronite bone adhesive perform equally well under both gravity conditions. Histologically, the tetranite-treated defect form woven bone, so immature bone, but even if it was immature, it was mineralized bone, as you can see from the bone costa in the histological images, without exceeding the anatomical boundaries, unlike BMP2 treated defect, which form excessive lamellar bone and expanded marrow space. Quantitative micro-CT analysis confirmed these findings. Tetranite and BMP2 both significantly outperformed the negative control, but BMP2 induced disproportionate bone growth, especially behind the defect zone, even reaching the contralateral side of the skull. In contrast, tetranite bone adhesive support controlled, localized bone regeneration, making it a promising candidate for safe bone repair in space and earth environments. So in summary, tetranite bone adhesive promote effective and spatially restricted bone regeneration under microgravity condition. And this is a key step toward the regenerative treatment in space medicine. So we can conclude that this investigation demonstrates that tetranite treatment can effectively lead to regeneration of critical size defect, and that can do that in a way that were more predictable than BMP2, both in space and on Earth. So you can say that if further studies in humans confirm these results that we obtain in animal studies, we have a novel biomaterial able to regenerate bone, and it can become available for future space explorations and for better health on Earth. And also, we can say that while previous studies demonstrate that microgravity can suppress stem cell differentiation and impair long bone healing, our findings challenge this assumption, because we have shown that with the right stimulation, calvarial bone regeneration is not only possible, but can be effective. This opens up new questions. Maybe there is other form of regeneration that can be enhanced with targeted approach, or maybe we can just reshape our strategies for long space travel or even terrestrial bone healing. So, I encourage you to focus on these, not just on the limitation of microgravity, but on the potential it holds when paired with the right biological interventions.
Hi, Roberta. Thank you for your talk.
Thank you. I realize now that I didn't record an audio for the last slide. So I want to just thank all the collaborators that help us, people from RevBio and all veterinarians from NASA, Leidos that, help us to complete this amazing project. So thank you all, and thank you for having me today.
Yeah, absolutely. I'm happy to have you. I guess while we're waiting for the Q&A to populate, I'll kick it off. So it's exciting that tetranite performed well in microgravity and on Earth and better than other methods of regeneration. Are there any other applications that you've considered using it for, or is it limited to bone regeneration?
I think that we can use for sure for other things. So the material was developed for bone regeneration, and the company tests this material in a lot of field, like dental field, orthopedic field. So there were a big number of applications. So they are still ongoing, this experiment, and I see that they have very promising results in different fields, not just regeneration, but we are still waiting for the results from the company.
Okay. We have several comments in here about great presentation and how important this topic is. Do you know what the time for complete replacement of the tetranite may be in space, or will it not be completely replaced?
Okay, so this is a very important question. Thanks for that. Actually, the mice stay in space 60 days. So when we received the samples after 60 days, we were not sure if the material was completely resorbed or not. But when I start to cut all the samples, and there were a lot, I can be 100% sure that the material was replaced, even because I tried to perform the same staining on a single disc of tetranite, and it's impossible to stain the material itself. So when I stain this bone tissue, I'm sure that what I see was just bone. there were no like material or like left on the side, on the defect. Maybe there was some like residual fragment, but it was very, very, very small in comparison to all the defect that we regenerated.
Okay. The next question, your study was done in mice, correct?
Yes.
But mice and humans aren't the same. So what differences would you expect to see in humans?
So actually the company is performing studies in the dental field in human. So, I mean, our experiment was on a critical size defect. We know that the mouse model was a very good model to study this. Of course, there are differences, but the mouse are like, genetically speaking, is like very similar to human. So what we did after this project, we compare the gene expression that we obtained, because together with this, there was another aim with other mice that stay 45 days in space. So I didn't present this data today, but we have other data on gene expression, and we compare the data obtained with some data that are online, so like available from human. We saw some similarities. We're still ongoing on this, so maybe next year I will present the genetic data, so we'll see which difference we have, but there are some similarities. Of course, there are differences because we have human and mice, but the next step will be maybe see if there are similarities, and maybe we can apply this material in human. Could be a good, like, results.
We have one person who'd like to know how they can reach you.
Yeah, of course.
Is your
Yeah,
contact information in the full presentation?
Yes, I just put I can even, put on the chat. So I work in Pittsburgh in the School of Dental Medicine with my PI is Dr. Giuseppe Intini, and I share the detail on the chat for sure.
Great, thank you.
Thank you.
We have another question here. How do you think this is going to be helpful for long duration space missions, such as to the moon or Mars?
Yeah, so actually that was our idea, because I mean, we know that microgravity impact musculoskeletal physiology, so it can occur that we have fraction in space, and this material is very easily applicable because we have like an injectable sealing, and you can easily applicable this material like a glue. Of course, we need to, like better study this aspect to see that could be available for long-term like space mission. And another important thing of this material is that you can use to fix like fracture and to help regeneration and to ameliorate like bone loss in case after the astronaut come back on Earth. So there were very different applications that you can, study in the future.
Okay, so I think that answered the next question well, of why you decided to test your product in space. The combination of long-term space travel and then also bone muscle density loss.
Yeah, even because the amazing results was that, in the last report that I saw, we know that microgravity can impact stem cells, so under microgravity condition. So we were thinking, we expect that, and the results was shown in the negative control, we didn't have any regeneration if you didn't use any help. So it looks that this material can improve the regeneration, helping the skeletal stem cells to differentiate, so to revert the quiescence that the microgravity can lead to. So, we need to better investigation this aspect, but we have promising, expectation on this.
Okay, great. And we have a couple more minutes for questions if anybody has anything else. If not, what do you think, Jim? Can we take questions from other speakers still online, or?
No, I think there's one more just popped up there. So just ask that one, and then we'll take a break till the next session.
All right. So our last one here, and we have a couple more asking for your contact information, so you put that in the chat after.
Sure.
But you mentioned use in dentistry.
Yes.
Do you mean that you would use this instead of a cadaver bone?
Actually, I'm not sure about that. So we should ask the people from company if they did, but they use in dental field. I know that they performed some experiment in Italy, in other country. They obtained, the possibility to do that. So they're working on different fields.
Okay, great. Well, thank you so much, Roberta.
Thank you. Thank you so
It's
much.
great to have you, and we enjoyed your talk. And
Thank
we
you.
look forward to hearing more next time.
Of course. Thank you so much. Have a good day.
All right, you too. Well, that concludes our session. I think we had a lot of great presenters and a lot of great results. I come from a physical sciences background, so I'm kind of just learning the biological sciences side, and it's been very interesting. I can say that you all definitely have harder experiments and also harder jargon to pronounce, but I did my best. So thank you all, attendees and authors, for your time and your interest in the space industry. Just a reminder that all these presentations will be available online, and the webinars are being recorded and will also be available online as soon as possible when we're done here. Our next session of the day will be Human Health and Performance from 10:25 to 11:35 a.m. Eastern Time. With that, I'll let Jim give a quick closeout.
No, I got nothing else. Yeah, we'll pause the webinar. You guys can stay in if you want to. Yeah, and we'll be back here at 10:25.
All right.
Thanks so much, Philip.
Yep. Thank you.
Thanks.